Welcome everyone today to our Optimism and Innovation talk titled Old Drug New Tricks, Thalidomide's Journey Through the Ages. Today we are joined by Dr. Katherine Donovan. Katherine Donovan is a senior scientist in the Fischer Lab where she works on the development of a new type of therapeutic and tool molecule for targeted protein degradation. Today Kat will be speaking about her journey to Dana-Farber as well as the 65-year journey of thalidomide from exile to FDA approval and what lies ahead for this enigmatic drug.
Today, my talk title is Old Drug New Tricks, Thalidomide's Journey Through Time. And this is me. My name is Katherine Donovan, and I'm currently a senior scientist working at Dana-Farber Cancer Institute in Boston, Massachusetts. So today I'm going to start by talking you through my career journey, and then later I will talk to you about the journey of the infamous drug called thalidomide. So I was born in Christchurch and grew up on a small farm in Rangiora, which is about 30 minutes north of Christchurch. I don't have an academic family at all, so I think my interest and drive to follow the scientific path that I have taken has mostly come from a curiosity that I have within. So I'm the oldest of 5 children. And growing up, we spent most of our free time outside exploring and building things and climbing trees. I think perhaps that environment of freedom to explore and experiment really helped to nurture my curiosity with the natural world and how the curiosity I have to understand how things work. So through my early schooling, I definitely gained an interest in studying, and I really loved to read. But it wasn't until high school that my interest in science really started to develop. I was always really looking forward to and excited by biology and chemistry classes. I found the content really, really interesting. I enjoyed the homework and quizzing myself, and I really loved the intellectual challenge that science brought. So the struggle to understand a concept and then that feeling of achievement when it finally clicks. So through the early years of schooling to undergrad, there are many people who have helped and supported me along the way, but the names and faces on this slide are of those who have made the biggest impact on my early academic years. So I was incredibly fortunate to have and still have the support and guidance and knowledge from all of these amazing mentors. So I started working as a waitress a couple of days a week after school and on weekends when I was about 14. I like to be independent and I like to keep busy, but also with 4 younger siblings, getting out of the house was always a really big bonus. I had a lot of fun working and I met a lot of great people. And surprisingly, I learned a lot of soft skills that really helped me massively in my scientific career. So probably the one that I think that has been the most valuable is the ability to network and talk to people. So an early example of this helping me was when I received a job opportunity from one of my customers at the restaurant I was working at. So this was near the end of my bachelor's degree, and while waiting tables, I was chatting, as I usually do, to a big group of people that looked like they were having a business dinner. So they were asking what I do and what I'm studying, and I told them that I was studying biochemistry, and they ended up asking me to come in for an interview for a summer research assistant role. So of course I followed up on this the next day, and I ended up leaving my job as a waitress and spending my summer as a research assistant at Ansco Foods, where we were researching different ways that we can use the remains of meat products to make healthcare ingredients. Then near the end of this summer, I was really torn between going back to uni to do postgrad or getting a real job. And I think at this time, it really felt like a critical decision point for me because most of the jobs that I saw advertised were for bachelor level positions. And I realized that doing higher education may actually make me too qualified and harder to get a job in New Zealand. But in the end, I decided not to worry about the end goal situation and just do what I thought I would enjoy the most. And so I enrolled for a master's degree at University of Canterbury. During the next year, I had a lot of fun in my 4th year classes. I especially love protein biochemistry and animal physiology classes. And near the end of the year, I received a summer scholarship opportunity to work with Ren Dobson, who was at that time a newly appointed faculty at University of Canterbury. So I spent a couple of months working in Melbourne at Bio 21 with Ren while he was finishing up his postdoctoral work. So during this time while I was working with Ren, he actually offered me a master's scholarship to do the research part of my master's in his lab at the University of Canterbury, and that would have a focus on protein biochemistry. Within 6 months of joining Ren's lab, I had fallen in love with research and decided I wanted to continue my project beyond the 18-month time limit that they set on master's degrees. So this meant that I would need to either do a PhD after my master's or turn my master's into a PhD, which would give me a total of 3 to 4 years of research time in Ren's lab. So I spoke to my advisors at the time, and they agreed that we could turn my master's into a PhD, which I was really excited about. That meant I got another few years of studying and research and learning about lab skills. So my time in Ren's lab was nothing short of amazing. The lab was extremely happy, enthusiastic, open environment where there were no stupid questions, and we were really encouraged to explore and try new things. But like all of the best things in life, the PhD really requires a lot of hard work. Research is hard. It takes a lot of determination and persistence to turn up to work every day when everything you do fails over and over and over again. But I think when it finally works, and it usually does work, you get the greatest feeling of achievement. And I think that's something that really drives me in science and continuing to do science is I enjoy the struggle, and I really enjoy the achievement at the end of it. So a couple of years into my PhD, I had some questions that I wasn't able to answer with the tools that I had available to me at University of Canterbury. So I connected with a professor in Canada, Dr. Derek Wilson, and he invited me to spend some time in his lab doing some experiments that I really wanted to do. So I applied for it and I received funding to fly myself to Toronto, Canada, and spend a few months working in Derek's lab. So I had a mile-long list of experiments I wanted to do in a very short amount of time that I was in Derek's lab. So there were many, many all-nighters in the lab, but in the end, the sleepless nights paid off as I completed almost everything on my list, which I was really happy about. And these were the last experiments I needed to do or wanted to do for my thesis before I could move on and get a real job. So in addition to learning a lot of cool new science methods, I had a lot of fun while in Canada. So I connected with all of Derek's lab members immediately. I got to do a lot of travel around the Canadian East Coast, which is really beautiful. I got to give a talk, it was my first international talk in Ottawa, Canada. So that was a really cool opportunity. And then near the end of 2015, I was wrapping up my thesis and trying to figure out what I wanted to do next. I knew for sure that I wanted to move overseas and experience living elsewhere for a while. So that experience in Canada and Melbourne really, I guess, set in place the idea of moving overseas. I really enjoyed my time in traveling. I was also feeling a really strong pull towards continuing research, but I wanted to change my research field into something more medically related, such as studying proteins that are involved in causing different diseases. So I applied for postdoc positions, which are post-PhD training positions in the United States, Sweden, and Canada. And in the end, I moved halfway around the world to the United States, to Boston. So I joined Eric Fischer's lab at Dana-Farber Cancer Institute in Boston in March 2016 to study proteins involved in diseases and different ways that we can drug these proteins to treat diseases. I had only asked for a 1-year visa because I was very, very certain that I would only stay in the US for 1 year. And I wanted to stay for 1 year to learn new methods and gain a bit of experience in a new environment. And then I had planned to move home to get a permanent job in New Zealand. So that first year was extremely hard, probably one of the hardest years I've had. I moved to Boston by myself. I didn't know anyone in Boston. The experiments and the projects were really new to me. It was just a completely new environment. I was one of the first people to join the lab, so Eric was a new PI. So that meant that I didn't have a group of automatic friends or people that were already here that had to be my friend. But near the end of the first year, I had learnt a ton and had started making friends and finding my place here. And I decided I wasn't ready to go home yet because I felt like there was still so much learning to do and growing to do with Eric as a mentor. So after a couple of years as a postdoc in Eric's lab, Eric and Nathaniel Gray set up a Center for Protein Degradation, which I'll explain a little bit more later. And this was set up at Dana-Farber. So it's kind of like a biotech company within the institute. So I was promoted into a permanent role as a research scientist. And for the next 15 months, I worked split between being a scientist in the Fischer Lab and being a group leader in the center. This was an amazing opportunity for me and allowed me to learn about drug development and how to move a drug closer to clinical trials, as well as about budgets and hiring and managing a group. But working 2 roles wasn't sustainable long term. So at the beginning of this year, I made the very difficult decision to go back to the Fischer Lab full time as a senior scientist. The Fischer Lab has now grown to 20 to 25 amazing people, very, very talented people from all over the world. We have a really great open environment where we're all here to help each other succeed. No 2 days are ever the same in the lab. And we're working on solving some really cool problems, one of which I'm going to talk to you about next. So thalidomide. Thalidomide is a pretty remarkable drug, and I wanted to share its long and bumpy path with you today. So the DNA that we inherit from our parents codes for proteins, and proteins are these amazing molecules that do all of the work in our cells. Some of them turn food into energy, some recognize and fight off bacteria and viruses, some control the fluid balance in our cells, some are responsible for determining our hair, our skin, our eye coloring, and others are hormones or control or regulate hormones. So in our cells, our DNA is constantly mutating, and this can cause our cells to make abnormal proteins like pictured here. Fortunately, our cells have different repair mechanisms to fix this mutated DNA, or disposal systems in place which remove these abnormal proteins to help us have a healthy and normal balance. When our cells lose balance, like I've pictured here, of normal to abnormal proteins, this can cause our cells to lose control, and this creates an opportunity for diseases and viruses to take over. In the Fischer Lab, we study proteins called E3 ligases. Their role in the cell is to help maintain a healthy balance of proteins, and they do this by identifying and tagging unwanted proteins that need to be removed from the cell. So here we have a schematic, which is a drawing of the cereblon E3 ligase complex. So each of these is a protein. This is a network of proteins that work together to remove unwanted proteins from the cell. So cereblon is here in green, is responsible for deciding which proteins are going to be removed from the cell. And it does this by binding to the target that it wants to remove. So here we see our target that we want to remove from the cell. It's bound to cereblon. Once cereblon binds to this target and pulls it in, these smaller proteins that are red here, they're called ubiquitin, these get transferred onto the target. And these ubiquitins act as a degradation or disposal signal. It's kind of like sticking a trash sticker on something you want to get rid of. So this disposal signal here is recognized by another big protein complex called the proteasome. And the proteasome takes up these targets and it degrades or chops it up into pieces, like I've tried to picture here, and this removes it from the cell. So this is a really great imagery for what is happening. So this proteasome here is using its magnifying glass to look for this disposal signal, which is this red chain. And then it picks up these targets and chews them up and basically into little pieces that are gone from the cell. So basically our cells have this amazing disposal signal built in to remove proteins that we don't want in the cells. So here's a side-by-side comparison of the drawing or the schematic that I just showed you of the cereblon ligase complex. And next to it is a real structure that was experimentally solved in the lab, actually by my boss. So I've tried to color them the same so you can see how different it is. So here we have cereblon in green, and this is what cereblon really looks like. And DDB1 is here in yellow. So in our lab, what we're trying to do is we're trying to solve these structures of these ligase complexes and then use these structures to design drugs that can bind to them and help us take control of the cell's disposal system so that we can remove proteins that cause diseases. So wouldn't it be great if we could just simply remove all of the proteins that cause diseases in humans? Unfortunately, it's not quite as simple as it sounds and we would like it to be. But I want to tell you about one of the drugs that can actually bind and take control of this ligase. And this drug is shown to be binding to the ligase here in red. You can kind of see it here. And its name is thalidomide. So this is thalidomide. In its early days, thalidomide caused a worldwide uproar. It became very well known for the damage and suffering that it caused, as pictured in all of these newspaper articles from all over the world. The suffering that thalidomide caused has been described by experts as the largest man-made medical disaster in modern history. So today I'm going to talk you through the journey of thalidomide from its early days of devastation to the current hope that we think it could have in fighting cancer and diseases. So let's start back at the beginning. In the 1950s, it was a really rough time all over the world. It was post-war era. Anxiety levels were really high. People were struggling to sleep, which meant there was a really high demand for safe sedatives to help people get through their day and to sleep. Thalidomide was developed as a new sedative in the early 1950s. Initial studies were done on mice, and they showed that it was almost impossible to reach a lethal dose. This means that scientists could give mice really, really high amounts of this drug, and they found no change to the mice at all. So they were completely fine. They weren't able to kill them. So at this point in time, if a drug was safe for mice, then it was thought that it would also be safe for humans. Back then, there were not strict drug guidelines and controls like there are with new drugs now. So in 1957, thalidomide was marketed for sale in Germany. And then over the next few years, thalidomide became increasingly popular and was picked up by 14 different drug companies and marketed in over 45 countries around the world. So this advertisement is from the UK. Distaval is the UK version of thalidomide. You probably cannot read the fine print, but it says it's free from untoward side effects and is particularly suitable for children and the aged. So it was really marketed as a super, super safe drug. By the late 1950s, thalidomide was now also being prescribed really heavily for morning sickness. And it was described as this wonder drug because of its safety and almost immediate morning sickness recovery after women took the drug. In the late 1950s, reports of babies born with birth defects started to appear across the world. But back then, computer documentation and data sharing was not easy. So communication between different doctors and medical professionals around the world was a lot harder and slower than it is today. So by 1961, doctors had started to notice a significant increase in the number of babies being born with severe birth defects. Then at roughly the same time, two doctors on opposite sides of the world, McBride in Australia and Lenz in Germany, connected the dots and realized that thalidomide, this new and amazing safe morning sickness drug, was actually responsible for these birth defects. So in 1962, thalidomide was taken off the shelves of pharmacies and doctors' offices and completely banned from the market. The drug is now classed as a teratogen. Unfortunately, by this point, the drug had been available and highly prescribed for a few years, and this meant there were already reports of over 10,000 babies born with severe birth defects, including shortened or missing arms and legs, which you can see in these pictures, as well as eye, ear, and internal organ issues. There were also a really large number of reports of miscarriages that were also thought to be caused by thalidomide. But after all of this devastation, if we fast forward several decades, thalidomide was taken off the shelves again and tested on new diseases, and it was found to have anti-cancer and anti-inflammatory properties. Then in 1998, it was approved for the treatment of leprosy. In the year 2000, thalidomide was also found to be effective at treating a type of blood cancer called multiple myeloma. And then in subsequent years, new versions of thalidomide were made to try and increase the cancer fighting properties and remove some of those birth defect properties. And this led us to new versions of the drug called lenalidomide and pomalidomide, which have very small changes, which I've colored in red, to the scaffold of the drug. And these drugs were approved over the next couple of years for the treatment of multiple myeloma, the blood cancer. So what's astounding at this point is that it's been over 50 years. Thalidomide went from treating morning sickness and causing severe birth defects to successfully treating cancer, but we still have absolutely no idea what the drug is doing in our cells and our body to cause both of these effects. But in 2010, it was found that thalidomide binds to a protein called cereblon, which I mentioned earlier. Then in 2014, it was found that these drugs cause two proteins called IKZF1 and IKZF3 to be degraded within the cell. So here I've shown a graph. The dark bands indicate the presence of these proteins. So when you increase the amount of drug in these cells, these proteins disappear. So these drugs are removing the proteins from the cell. Then in 2014, the structure I showed you previously was solved, which shows that thalidomide binds to the cereblon ligase. So with all of this new data in 2014, the mechanism of how these drugs fight cancer started to make sense. So these two proteins, IKZF1 and IKZF3, have a role in regulating white blood cells, which are necessary to protect the body from infections. So these proteins actually help the growth of multiple myeloma by reducing the body's immune response to infection. But when we treat cells with one of these drugs, they bind to this cereblon ligase and they act as a glue, and they pull these proteins in. So we get this complex with these, the cereblon pulling in this IKZF1 or IKZF3 through this glue. And this causes them to be tagged, as I showed you earlier, for degradation by the cell's disposal system. Now that the cell has lower levels of these proteins, this means that the immune system can fight the cancer and the growth of multiple myeloma will decrease. So this was huge progress in understanding these drugs. But at this point, it's 2015. It's been 60 years since thalidomide caused thousands of birth defects, and the drugs are being used to treat humans again. But we still don't understand how these drugs cause these severe birth defects in humans. So to try and identify how these drugs cause the birth defects in humans, we needed to know what the drug did in the cells of embryos. So different cell types make different proteins. For example, skin cells make a lot of proteins like collagen and melanin that give skin a nice structure and texture and color. And embryonic stem cells produce a lot of proteins necessary for the development of limbs and organs within the human embryo. So we decided to see what thalidomide actually did to human embryonic stem cells. So that we treated these cells. This is the picture of embryonic stem cells. We gave them some thalidomide to see what happens. So we extracted all of the protein from these cells, and then we measured the amount of protein in the cell after drug treatment. So this is an example of a graph that we produce when we do these experiments. So in this top quadrant, this is showing us the proteins that go up when we give it drug. And in the bottom, this is telling us which proteins go down when we treat with drug. So when we saw the data from these three drugs, thalidomide, lenalidomide, and pomalidomide, we saw that they all caused different proteins to go down. So you can see proteins down here. In particular, all three drugs caused a protein called SAL4 to go down. So we asked the question, what is SAL4? So Google told us that SAL4 is a protein essential for normal development in embryos, and in particular, it plays a major role in the development of limbs. So after 60 years of no explanation for the thalidomide birth defects, we thought maybe we were onto something here. So if the embryo needs a lot of SAL4 to grow limbs, and this drug, thalidomide, is causing the amount of SAL4 to go down, then maybe this could explain why these babies were born with such severe defects. So we did further tests, and we found that these drugs are able to completely degrade SAL4 after just 4 hours of drug treatment. So this red band is an indication for SAL4 being present. The bigger and thicker the band, the more there is there. So after 4 hours of drug treatment, we don't see any SAL4 left in these cells. We also wanted to see how long it takes the cells to make more SAL4 after it was degraded. So we treated cells with drug, and the SAL4 disappeared, and then we took away the drug, and we measured how long it took for that SAL4 level to recover. And we found that it took 48 hours for SAL4 levels to come back to normal in these human cells. So you can imagine if you're a human taking this drug and you have an embryo developing in you, if your SAL4 levels that are needed for the limbs to grow on these babies are gone, and it takes 2 days for the levels to come back, that could have severe consequences on the baby. So through many more experiments, we confirmed that thalidomide is binding to cereblon, and it's acting as a glue to pull in SAL4, and this is causing SAL4 to be have this degradation signal or disposal signal, and then it gets sent to the proteasome and it gets degraded. So with a bit more research and Googling, we realized that there is another disease that looks almost identical to thalidomide syndrome, and this is called Duane radial ray syndrome. So they both have arm and leg, shortened arm and legs, a lot of eye, ear, and internal organ defects. And these two diseases were so similar that doctors have actually struggled to tell the difference between patients with Duane radial ray syndrome or mutations or patients that were exposed to thalidomide when they were in the womb. So based on our research, we think that these two diseases look similar because they're caused by a loss of SAL4. In thalidomide syndrome, thalidomide causes SAL4, so SAL4 protein's on and completely normal, but it causes SAL4 to be degraded, so there isn't enough left for the body to develop normally. In mutations, like Duane radial ray syndrome, the SAL4 protein is mutated, or SAL4 gene is mutated, and this causes the protein to be abnormal, so it doesn't function normally. It's not able to do its normal job of helping normal development. So essentially, both of these diseases, or thalidomide syndrome and Duane radial ray syndrome, We think they're both occurring due to a loss of this protein, which is essential for limb development. So this was all extremely exciting for us. After 60 years, we felt like we were finally starting to understand how this drug caused so much devastation around the world. But we still didn't understand one key thing, and that was, why does it cause defects in humans, but it doesn't cause defects in mice? Because mouse embryos also need the cell-4 protein in order to develop normally. So we did some experiments to test human and mouse cells side by side, and we found that thalidomide causes cell-4 to be degraded in human cells, but in mouse cells, it's not degraded at all. So after doing some more experiments, we found that the mouse and human proteins have some small differences around the position where the thalidomide drug binds. So in humans, the drug binds to cereblon as pictured here, and then this acts as a glue and it sticks to the cell-4 protein, which allows it to be tagged and sent for degradation. But in the mouse proteins, there are some small differences around these drug binding areas that prevent the mouse cell-4 from sticking to this glue. So the mouse cell-4 doesn't get degraded, which means there are normal levels in the cells, which means the mouse embryo can develop normally. So if we model that with an animation, so here we have thalidomide, it's binding to cereblon, which is red. The cell-4 comes in and it sticks to the glue. The degradation signal gets transferred to the cell-4, and then this protein is removed from the cells. In mouse, there are changes here and changes on this side that prevent these two proteins from coming together, which means that in the mouse, this protein doesn't get degraded and everything can develop normally. So I guess what we have found through the last 10 years of thalidomide's journey is that thalidomide acts as a glue to degrade different proteins in the cell. And this can be good or bad. It is good when someone's fighting cancer and is able to degrade and destroy the proteins that are helping the cancer to grow and spread, like IKZF1 and IKZF3. But it's bad when a baby is developing and the drug degrades and destroys a protein that is essential for the baby to develop normally, like cell-4. So thalidomide's journey doesn't stop there. It's actually being used in a new type of drug development. I wanted to touch really quick on these new molecules that we are working on in our lab and a lot of other labs around the world. The first versions have actually gone into clinical trials last year. So in 2015, it was discovered that we can choose and specifically target proteins that we know cause diseases and send them to be degraded. And we can do this by making a drug that looks a bit like a dumbbell. It's called a proteomics. So on one end of this dumbbell, we have the thalidomide, and it's binding to the E3 ligase. And on the other end of the dumbbell, we have another drug that's binding to our target that we believe is causing diseases. So we link these together through a linker like this, and they end up looking like a dumbbell. One drug on one end and one drug on another end, and then this linker in between to connect them. So when we look at that in terms of what is happening in our E3 ligase, what we have is the thalidomide's binding to the cereblon, and then the other drug is binding to our target of interest. Let's say this target causes breast cancer. And because it's being pulled in, this degradation signal is able to be transferred onto it, and then this protein is removed from the cell. It's degraded by the proteasome system and removed from the cell. So one of the ways that these dumbbell degraders or proteacs are different to current drugs that we have available now and that we take when we're sick is because they act differently. So normal drugs work by binding to a target or a protein that may cause an issue within our bodies, and it turns them off. So we have our target that's on, we have our drug, it binds to our target, and it turns that protein off. One of the issues with this is you need to keep a high amount of that drug in your body and in your system to keep that protein turned off. And this can cause a lot of unwanted side effects when we have a lot of drugs in our system. So I guess it's kind of like turning off a light switch, but to keep it off, you have to hold your finger on the switch. But degraders act really differently. They don't just turn the target off, but they also degrade and dispose of it, remove it from the cell, so that the amount of that target protein stays very low in the cells until the cell is able to make more. So we think this method of drugging different diseases could mean that less drug is required to see a similar treatment response as we see with typical drugs now. So just like thalidomide, I have had many, many ups and downs in my scientific journey. And every day I'm incredibly grateful for all of those people who have supported and mentored and shared their experiences and advice, and also just stuck by my side through all of the great and bad times. I want to thank all of these wonderful people. Also thank my bosses over the years, as well as all of the lab members who have just made it really easy for me to come back to work every day and do what I love. And also thank you all for listening. If you're interested in hearing more about science as a career, or learning about thalidomide or any of the projects that we have going on in our lab, I would be really happy to hear from you.
Thanks for sharing your journey with us, Kat. And I know I've learned so much in such a small amount of time, and as a visual learner I really appreciated those animations, so thank you for doing those. And you did mention that you had some really great mentors that have assisted in your journey, and more specifically though, what did those role models do to make you, I suppose, more passionate or get you passionate about science?
I think one of the big things that helped with the mentors and keeping me excited about science, and I guess keeping me coming back to work every day when it's hard, is their belief in me was a really big thing. Also just having mentors who are extremely excited about what you're doing gets you excited. So when my boss is like emailing me about data or getting excited about results and they're feeling it too, it really helps, I guess, pass that excitement down to me, and I guess the same with my other lab members.
It was wonderful to hear about your childhood and where you grew up, and you mentioned that you had the ability to have freedom to explore as a kid. Do you have a favourite memory that you have of exploring as a kid that might link to your exploration in science?
I spoke to my parents about this when I was preparing my slides, like why or how did I end up a scientist, because it just doesn't make sense with my family. I think it's really hard. I don't think there's any particular thing that really drove me to it. I think just being outside and not being forced in any direction that my parents may have wanted me to go in just really helped me find what I wanted to do. So my dad said to me, he said, "I don't think you chose science, I think science chose you." And that's really all. I really don't know where it came from. It doesn't make sense, but it happened. Yeah.
Yeah, that's awesome. And sometimes things just work out, hey, just meant to be. So maybe it was one of those sort of things. So you mentioned that you did some work in high school, and that those experiences or journeys contributed to you learning those soft skills or enterprise skills through being able to network and communicate. What's one tip you would give someone who's looking at a career in science and how they could improve these skills?
Definitely talk to people. It's something I tell a lot of the people who work with me who are going on to do PhDs. It's one of the biggest, I think it's really, no one really emphasizes it ever. And it's not until you need a job or you have a reason to reach out that people realize they don't really know how to do those things. So I think not being afraid to contact people. I remember when I was writing essays in undergrad and I would contact these big famous scientists, and my friends were like, what are you doing? They're never going to reply to you. I'm like, well, I can try. And usually they did. It's actually a really big compliment to them to have someone contact them and ask about their research. Like, we love that. So I think not being afraid to reach out to people and talk to people. So I talk to everyone at work, from the cleaners to the salespeople to everyone. And it's amazing having that network and having so many people who are around science and involved in science. They may not be directly involved in your day-to-day, but I think that's really, really important. And I wish they taught us that more. I remember my PhD advisor used to make us, we would have big speakers come in from overseas, and he would make us drive to the airport and pick them up. And it was like 15 minutes of, oh my gosh, what am I going to say to them? I don't have, like, I'm just a student. And I think that really helped develop those skills as well, where we were kind of put into these uncomfortable situations and you had to learn to adapt to it, I guess. And that's been massive help for me.
I think that's really wonderful advice. And I know that any student that listens to that might start to think about who they might be able to reach out to, as opposed to not reaching out at all, which I think is really exciting. Just back on your career there, what is the most fascinating thing you've come across in your career, or what is a wow moment? I mean, everything you've mentioned so far seems pretty wow to both of us, but what's something that stands out to you?
I think there are different things. So I think scientifically, discovering cell 4 was just something I really wanted to do, but I never thought it was possible, because 60 years and no one's figured this out. And there's so many people even now still suffering from this, and they still don't have questions or answers or anything. So I think publishing that work was amazing. It's not finished, and it's something that will go on for a long time because science is slow, but I think at least giving people some sort of answer was really important to me. Another wow moment is just having people contact me. So I've had some people who were born with these thalidomide defects reach out to me on Twitter and email, and that's really nice to know that even if it's a small thing, they are getting some answers and it's helping them, I wouldn't say come to peace with it, but get some answers, I guess.
So Kat, we just want to thank you again for joining us for the Optimism and Innovation Talks for the Bendigo Invention and Innovation Festival. And I'm sure I can talk on behalf of us, but we have enjoyed listening and learning. I've definitely learned a lot, and I really hope those that have registered for the session have learned a lot. And thank you again for your time.
Awesome. Thank you so much for inviting me.